So the final speaker of the session is Sampath Perumal. He completed his PhD in South Korea and then has been in Saskatoon ever since. He did a postdoc with Agriculture and Food Canada where he was doing genome sequencing of Brassica species. And he's moved on to work with Andrew Sharp and myself on analyzing structural variants in canola.
Thanks, Isabel, for the nice introduction. It's my first presentation after COVID in person as well. So I'm nervous, as you see me. But however, so this is an ongoing project, and I'm going to introduce our project. This is to provide an overview of structural variant landscape in Brassica napus genome. My name is Sampath, and this presentation is using NAM population. We have 50 NAM founder lines, so we characterizing structural variants to get more novel genetic variants to tackle complex traits. So outline of my presentation goes like this. So I'm gonna give a little bit of introduction about structural variants, and then the crop itself, the canola, which is important for this province as well as the NAM population, and then the long-read sequencing approaches and identification of structural variants, and then some future prospects. Okay, what is structural variants? Structural variants are larger genetic rearrangements, typically less 30 base pair or 50 base pair to few kb to MB sizes. These are, you might be familiar with the single nucleotide variants. However, these are different types of structural variants. This is really hard to predict using, or more accurately predicted using long-read sequencing technology rather than short-read sequence technology. These are different types of things. Like they can be different sizes. These can go up to few kb to few MB size. So this has real influence on genes. For example, if they have many copies, that can influence the copy number variation and then expression differences, something like that. So as you see that there are different kinds of structural variants, which is being poorly characterized using that short-read sequencing technology. And we are using the long-read sequencing technology to characterize these structural variants because, as I said, it's impacted not only the gene structure and the expression pattern, and also it has produced a lot of important novel variants, which helps to identify or tackle the complex variants. So we are exploring these hidden variants in the napus genome. So one quick example here. This is one of the structural variants work in Ardopsis, where it shows that these are the phenotype associations are predicted with structural variants which are failed to predict using SNP markers alone. So it definitely you need to work on, or you need to identify those structural variants which is impacting the phenotypes of interest, such as yield or resistance. Here again, I like to show that how good that long-read sequencing. There are a lot of information to digest here, but I just like to focus on this green dots here. These are this is the plot showing this structure variants identification with much better accuracy in the long reads. So you see that this long read based structure variant recalling rates are really high, and it's quite efficient compared to this short read one. It's in the orange. That's what we are trying to exploit in the NAM population or the canola lines. It's not only just identifying that accurate, it's also identify more number of structural variants. For example, so up to like 8 times in this particular Ardopsis crop has been identified when you use the long read sequence technology rather than short reads. The crop, so the canola is one of the important crop for the province, and it's a complex crop by itself. It's a allo tetraploid. This is the triangle of U. showing the genetic relations of 6 different Brassica species. This canola, it is formed by polyploidization between Brassica rapa and Oleracea, forming allotetraploids. This allotetraplod, it's about like 1 giga size genome, comparatively a good genome to work with, considering the scenario now. However, this has its own complexity, and it also has lack of diversity. It has lots of homologous exchanges happening, and in addition, there is a lack of high linkage disequilibrium, suggesting that's the lack of genetic diversity due to its recent origin, as well as that selective inbreeding for the traits such as disease resistance or that yields. So there is a need to increase the genetic diversity of these lines to get the full potential to tackle the complex traits as well. For that, Isabel and team, they have developed a NAM population where you just cross on, where you cross a light line against different genotype of interest. Having here they used 50 diverse genotypes crossed against a light line which is called a NAM0. It's a kind of a schema where you used 10 lines to cross against a founder line to develop a NAM population. So likewise, they developed about 2,600 recombinant inbred lines with 50 NAM families or real families. So this is the lines we are using for this to characterize the structure variants. So the top PCA plot showing that the diversity of those 50 lines. So this is the summary of the nanopore data developed using those 50 lines. So this is developed using Oxford nanopore technology at the Opal platform. They have this excellent data developed. Even like we exceeded almost like 150. This is mostly from single Promethean flow cell, but some of them are topped up with being topped up with Minion flow cells as well. And you see that the read lengths, so over 20K size reads, are really high. We are just produce this data to at least minimum of 50x coverage, not only for the purpose of structure variant, but also to develop a reference quality genomes. So that's why we want to have like high coverage of genome developed. And more than that, you can use the same data to call methylome as well. So it's quite interesting. That's why this long read sequencing can be exploited or explored in that manner. So our structure variant calling pipeline, we used at least 50x coverage. We had enough coverage because we developed for our whole genome assembly purpose as well. So we had this enough coverage, but you can still go with 30x coverage, or even 25-30x is should be good enough. And we used N99 as a reference genome, which is one of the found elite line, which is the elite line in NAM0 used and mapped against them using long read noise C caller. So we used multiple different callers to call structure variants, and then we filter out based on size and then read depth, and also consensus call approach was used to develop this pan structure variants. So here's the summary of total number of SVs identified from each different NAM lines. We identified at least 50,000 structure variants to 100,000 structure variants. There are 5 different groups among these 50 lines: Australian, Canadian, European, and the exotic, mostly from spring types, though we also include some winter types as well. So you can see that there are number of structure variants are really high in this exotic lines. These are the variants that need to be exploited. Like this is already in the pool, so in the real population, so that can be so that gives more advantage of increasing the genetic diversity and also to tackle the complex traits. This is just to show the glimpse of what's the size of the structure variants looks like. As I shown in the very first slide, so structure variants can be really bigger size. So here, the mid ranges like about like 100 to 1,000 base pairs to like 10,000 base pairs are really high. So this is definitely can't precisely identify using this short read or the Illumina sequencing technology, though you can get number of structure variants, but you can't get all the variants using the short reads. And the deletions insertions, obviously as expected, it's quite high in the genome compared to the other different types of structure variant. This is to quickly show that how we validate the structure variant using the read depth approach, or this is the IGV based integrated genome viewer visualization, where you see that redistribution on top. You see the deletion identified using the different colors, where that 1,338 base pair is clearly showing identified in based on the read distribution. Based on different random calls identified, we could able to say that so over 90% of the calls can be more accurate in this approach. Though we have to do that manual validation for some of the calls that we can't able to clearly validate using this read depth approach itself. This is a circular plot showing the distribution of different types, all the structure variants. So one of the interesting phenomena in this is, so here we show that distribution of the structure variants in all the chromosomes of the NAM0. So you see that the top, these red ones are the gene density. The red is the more the red is the more genes, and the white ones are the less genes. So you see that the structure variants have quite distributed in the genic regions or the euchromatic regions the most. So that's a quite interesting thing. That's already saying that it's need to be explored in the genome as well. And here it shows. Here we show the distribution of the SVs in the genes. The another interesting factor is that is already see that it has that biased association with euchromatic regions, but we also see that over 15 to 20% of the structure variants are in genes, and about 50% of them present within the 1 kb of this genes, suggesting it's definitely there is a impact of the structure variants on the gene structure or the functions. Before my summary slide, so this one is showing 1% of the active transposable elements which are contributing these structure variants. So summarizing my talk, so we used long-read sequencing technology to characterize or identify structure variants more accurately. We identified 50,000 to 100,000 of novel variants in the Brassica napus genome NAM population, which is going to be really important or boost for targeting these complex traits. And we found that the mid ranges like 50 to 1,000 kbs are really high in the genomes, and over 15% of them in the genes. So that also is quite interesting factor. We are still working on with the gene expression or like other association analysis as well. As I said, like it's early stage, but it's quite interesting results already. And this pan genome SVs is going to be definitely helpful for the genome and to tackle these complex traits. Thank you very much for your kind attention. Thanks.